Welcome back to this course on organic chemistry and biology and drug development. In the last session, we were discussing about combinatorial chemistry, that is a technology which has been developed to produce a large library of compounds and then testing it through high throughput screening, and the whole exercise is done to have a quick access to the heat compound, so that the heat compound can be then lead to lead and then lead optimization. Okay. So, this process is usually earlier days it was taking lot of time because the biological screening was taking a lot of time. So, the synthesis even if you make few compounds per day or per week, that would have been sufficient for testing within the time frame by the biologists. Now, because of the advent of high throughput screening, that there is a demand that you produce lot of compounds in a particular day or two, and then get it tested as quickly as possible, because the whole idea of medicinal chemistry or drug development is that one should, as there is failure, there is a high risk of failure, that one should really very quickly know what are the failure compounds which are going to fail or which are failing in the high throughput screening, and try to pick out the lead compounds or the heat compounds as early as possible without wasting much time and energy. Okay. So, towards that end, this combinatorial chemistry was developed. This is basically a synthetic technology in which a large library of compounds are made. Basically, there are two techniques. One is called parallel synthesis, and this slide what is now shown here, I have already shown it to you, that in parallel synthesis, each well, this is basically a 96 well plate, each well there is a bead, there are few beads, and then you have produced a particular type of compound. It is not that in a particular well you have a mixture of compounds. Okay. There are several beads, but all the beads are connected to, suppose if I take this groove, if I take this groove will have ultimately in the final product, which is represented by Z1, but Z1 is basically a combination of X1, Y1, and that is, and then you are adding this, making the hidden tones. It is a 3-step, basically it is a 2-step. The first step is that adding the Y1 to the X1, and then you do some reaction, you apply some reaction condition like heating with hot 6 molar HCl. So, that this is a particular type of combinatorial chemistry where you are making hidden tones. Okay. And these hidden tones, what I am trying to say, that each well has only a defined compound and you know what is the structure of that compound. On the other hand, if you go to the other technology which is called mix and split, in this case you get a larger library of compounds, much larger library of compounds, but the problem is that in a particular well, or if you are doing it in test tubes or your container, we will have beads where each bead is connected to a particular compound, but you do not know that it is a collection of beads and it is a collection of different compounds, but only one thing you know, that each bead is only connected to one particular type of compound, but you do not know which bead it is. Okay. So, let us start from there where we ended. So, in this mix and split, you take the resin bead attached to the functionality, and then you divide it into 3 pots, and in this pot you add A, and in the other pot you add B, and in the in the third pot you add C. So, you have resin bead here attached to A, resin bead B, and here resin bead is attached to C. But then you mix these two, take all them together and then split. So, what will happen, that in each of these beads in a particular well or test tube, it will have A, it will have attachment to B, and it will have also attachment with C. So, that means there are beads which are attached to A, there are beads which are attached to B, there are beads which are attached to C. Okay, now you add, you basically you have distributed into three pots or 3 wells, and you are adding D in the first pot, E in the second, and F in the third. So, in the process what you are doing, you are getting here A, D, B, D and C, D. Here you will get A, E, B, E, C and so on. So, in the third one, like you are adding F, so A, F, B, F and C, F. Then you again mix it and split. So, when you split, your each well will have beads which are connected to A, D, B, D, C, D, A, E, B, E, C, E as well as A, F, B, F and C, F. Now, what you are doing, you have again split it into three pots and then add G. So, when in one pot you are adding G, in the other pot you are adding H, and in the third one you are adding I. So, what will happen here? There will be 9 different compounds attached to the beads, attached to separate beads. You must understand this, that a particular bead will not have say A, D, G and also it is attached to B, D, G. That will not happen, because you are covering the functionality, whatever number of functional groups attached to the bead, that are all attached to either A or B or C when you started the synthesis. So, each bead is connected to one type of compound, not it is a mixture of compound. Okay. So, now you have 9 compounds here attached to the beads, here also 9 compounds and here also 9 compounds. Now, what you do? You test these beads containing the compounds and see if there is any bioactivity in any one of these wells or pots wherever you are doing the reaction. Suppose there is some activity shown by this cluster. Okay. So, the next thing is that you do not know actually which bead is connected to what, but what you know is that out of these beads at least some compounds are bioactive. Okay. So, the problem now is to basically to know what is the compound attached to a particular bead. See, you can individually separate these beads and also test it, test against bioactivity, and then suppose I get a bead which shows bioactivity, but I do not know what is attached here, whether it is ADG or BDG or CDG. Okay. So, how to do that? Now, you can say that I will take the bead and then detach whatever compounds are there. Remember, one bead does not have only one valency, it is a polyvalent bead. That means, you had from one bead you can get several molecules of this, okay, but all are same compound. If it is ADG, although I do not know when I take the bead, so all are ADG here. So, there is no scrambling of the structure of the compound that is attached to a particular bead. But the question is, the big question is how to know what is the compound that is attached to a particular bead, because these beads are not colored. It is not that blue beads are always having ADG, red beads are having other compounds, it is not that. Each bead is of same color, everything. Now, you have to basically deconvolute that you have a bead which is attached to a compound and that is showing some bioactivity. Now, the task is how to know what compound I have in the attached to the bead. Okay. How to do that? One way is that you break this bond between the bead and the terminal end and see the sequence of these different entities A, D, G or it is B, D, G. You can check that. Okay. But this if these are some compounds which are not very easy to do the sequencing. You know, sequencing can be done usually on peptides and as well as for nucleic acids. These are easier ones that you can do Edman degradation or you can do Sanger's method to know the sequencing. Other compounds, if it is different types of compound entities A, B, C are different entities, then it will be very difficult to really know what is the structure of the compound that is attached to a particular bead. So, how to know the structure of the compound attached to a bead? There is a technique which is called tagging technique. I will show you what is that. So, what you do, that each bead, suppose this is my bead which has shown some, no, this is the bead to start with. It is I say that these are polyvalent, that means there are a lot of binding sites here where I can add add A, B or C, that is my reacting partner. Okay. So, what I do, that when I have this bead, I have the bead attached to a linker, and that linker is distributed in two channels. One side you can do one type of reaction, and in the other side you can do the synthesis that is you are interested in. Maybe an example will demonstrate it, or you can, it will be easier to understand what I am saying is the tagging method. So, what is done? So, you have a bead, you can forget about the linker for the timing. So, the bead has reactive sites. In one reactive site you are adding your A, B, C, D, E, F, and on the other side you are adding something which is easy to sequence. Okay. Suppose I have two, in this case I just simplify it, I have separated the beads into two aliquots. Okay. And in one aliquot I added glycine. I am interested in making peptides. Okay. So, in one aliquot I added glycine. So, the glycine gets attached at the synthesis site, and at the same time I add some bases. In this case it is say CAC-ATG. So, I add a base, but for understanding you can say that I add something which is denoted as P1 okay, here, and here on the other aliquot I add methionine, a different amino acid, and I add another set of bases which is denoted by P2. So, what will happen, that this bead which is now can be represented, that it has got glycine here, and on the other side, because I am adding glycine and then also I am adding this P1 which is a combination of bases, the bases that are present in DNA. So, glycine, and on the other hand you are having this P1, and in this case in the other aliquot what you have methionine, and here you are adding the P2. Okay. P2 is in another collection of bases. Okay. So, then what you do, again you mix and split. If you mix and split, and then suppose I add again glycine here and glycine here. So, what I will get, because I will get in one particular container, I have two containers. So, when I mix and split, so here the beads will have both the characteristics, this as well as that, because I have mixed it and then splitted it. So, when I added glycine, what I will get is that I will have a bead, and remember, whenever I add glycine I add the same set of bases P1. Okay. So, what will happen now, this bead will have glycine, and then if it is I have added another glycine, and on the other side at the same time I add this P1. So, P1 will be attached to P1, and in the other bead what will happen, the bead which is in the same container, that will have glycine, and if it has reacted with sorry, it is not glycine because I have mixed it. So, I also have methionine here. So, methionine, and that will be attached to glycine, and in the tagging side, that means my P1 P2 side, what I will have here now, sorry, P2, because whenever I had methionine I had P2 there, and then P2 will be attached to P1. So, this is the scenario. Now, suppose I stop at the dipeptide, suppose I stop at the dipeptide stage. Okay. I see that one bead is giving some some activity. Okay. Then I am interested what I have added here, whether it is methionine glycine or it is glycine glycine. So, what I do, because I have this, I know that if it is methionine glycine I will have P2P1, and if it is glycine glycine I will have P1P1. So, I make the complementary base sequence and see which gets attached to this base sequence, whatever is represented by P2P1. Okay. Because that is what is called primer. That is why this name primer is given. What is primer? Primer is that if you have a sequence of bases, you already know that, and if you give the complementary base, then they are going to go and hybridize. Okay. So, if it is P1P1, you make a set of primer, one set of primer which is complementary to P1P1, and another set of primer which is complementary to P2P1. So, now you see that which primer is actually giving the hybridization, and then if you see that the complementary of P2P1 is hybridizing with the bead tagging site, then you know that it must be having methionine glycine. And once you know that, then you can separately make methionine glycine and do the bioassay, and you can vary. Now you know that the first amino acid has to be methionine, so you can take different methionine and then other amino acids you can vary and then optimize the heat. Okay. So, I hope this is clear. So, basically what you are doing, you are adding your synthesis, your compound which is required for the synthesis, at the same time you are adding a tagging entity. In this case, our example was a base sequence, a particular type of base sequence, because the DNAs are very easy to detect, means the base sequence is easy to detect by synthesizing the corresponding primers and see whether it is hybridizing or not. So, from that hybridization result we can tell that what is the tagging code, it is P1 P2P1 or it is P1 P1, and then you can or in the other test tubes what we will have, you are adding again methionine. So, it will be here it will be the tagging code will be, if it is glycine and then methionine. So, that will be P1 P2 and the other case is P2 P2. That means, when the bead is connected to glycine, I know that it is attached to P1 on the tagging site, and when I added the methionine, so that P1 will be attached to P2, because with methionine I add the P2, and then so you will get whenever there is glycine methionine, here it is methionine glycine, whenever there is glycine methionine, then the tagging sequence will be P1 P2, and whenever it is methionine methionine, the tagging sequence will be P2 P2. So, you see you can get 4 different tagging sequence attached to the bead, and from the tagging sequence you can tell what is the peptide sequence in the desired compound. Okay. So, that is now there are different techniques. These tagging techniques are various types. I will not go into very details, but I will show you another tagging technique. Instead of having this these nucleotides, you can bases, you can have other types of tagging systems, like in this case it is said that resin means the resin bead. So, if you the resin bead, you know that there is a synthesis site where you do the synthesis, and there is a tagging site where you do the tagging. Okay. So, whenever you add one component, you have to add the corresponding tag. Now, this tag, earlier I told you about the bases, nucleotide bases. Someone has devised this, that you can whenever there is a synthesis done, you on the other tagging site, what you are doing, you are adding this nitrobenzyl oxycarbonyl and attached to a linker, okay, and the linker is attached to an aromatic, a substituted aromatic moiety. Again I repeat, there is this the bead, and the bead has synthesis sites and bead has tagging sites. Okay. One more important point is that synthesis site is not just one, there may be several synthesis sites and there may be more tagging sites also. Basically, what you do when you do the synthesis, you maintain the concentration at such a level, so that the synthesis sites are more or less covered, leaving the tagging sites. Tagging sites, remember, they are also reactive entities. So, you have to be careful, the tagging sites are left free. Okay. So, whenever you do whenever you put again, suppose the synthesis site I put A1, okay, and in the tagging site I put this via this paranitro, this thing, the paranitro benzyl oxycarbonyl orthonitro, sorry, not para, orthonitro. This orthonitro is like the, I gave you the veratryl oxycarbonyl, which is photolabile. This orthonitro benzyl oxycarbonyl is also photolabile. So, if you shine light here, so what will happen, that this goes off, carbon dioxide is liberated, then you are actually generating ultimately the aryl halide. So, this is the mechanism that you form, this forms the aldehyde and this loses the carbon dioxide, and finally, this aryl tag comes out as the this is actually a benzyl system, CH2OH. Okay. So, aryl tag, we can simplify it. So, basically there is a linker which is photolabile, sorry, this is a no, this is not right, yeah, I hope, no, still this is red, I want to change the color. So, this is the photolabile group, and then you have this aromatic ring, this is no, aromatic, so this is A1, so this is AR1. Okay. Now, there are other tagging sites here, okay, and there are also synthesis site here. So, you maintain it in such a way that all the synthesis sites are blocked with A1, leaving, and the AR1, this one you maintain it such a concentration that some of the tagging sites are still vacant, remain vacant. So, whenever you are adding A1, you are adding this benzyl oxycarbonyl thing with a AR at the terminal end with a different AR1, aromatic aryl ring, okay, leaving some of the tagging sites still vacant. Okay. So, when you do the next reaction, so that means in the first reaction what you are doing, you are adding A1 and you are also adding AR1. AR1 means via this benzyl oxycarbonyl. Okay. And then when you take suppose A2, you are adding now A2. So, when you add A2, you add AR2. Okay. So, A2 will be attached to A1 here, all these A2 will be attached to A1, and this AR1 actually ends there, because AR does not react with another AR. So, what will happen, because some of the tagging sites are now free, vacant. So, now this will be connected to AR2. Still some tagging sites are left vacant, it is just a calibration of the concentration that will. So, this is little tricky, that you maintain the concentration in such a way that your synthesis sites are all filled up, okay. But your tagging sites, the tagging reactive sites are still free. So, one site is occupied by AR1 via this benzyl oxycarbonyl, and then the next site, adjacent site, you put a AR2 when you do the second reaction, and if you do a third reaction, you can put you can consider, because still some tagging sites are left empty. So, you can put AR3 also. Okay. So, that is the situation. Now, what is done here is that after the everything is done, you shine light, and when you shine light, your benzyl oxycarbonyl falls off. So, what you will get is the AR CH2, you will get AR1 CH2 OH, you will get AR1, and this is AR2 CH2 OH, then you will get you can get AR3 CH2 OH like that. Okay. Now, you do a HPLC, and if you do HPLC, what will happen? Each tag is different. So, each tag will show its profile, the chromatogram. Okay. And depending on the number of tags or number of peaks corresponding to different tags, you can identify what is the sequence of the of these entities A1, A2, A3, all these things. So, now let us see, I think this is still not clear, I want to make it a little bit clear. Now, in the bead, again this is a peptide synthesis. What is done is, this is the tagging. So, whenever you add glycine, so you have, these are tagging sites. So, you add T1. Okay. So, you add T1. So, basically you are adding glycine, so you are adding T1. Then if you add, because there are these beads are distributed suppose in 3 ports. So, in the first bead you are adding glycine and you are adding a tag T1. Okay. In the second container you are adding what is there, alanine, and you are adding the tag means representing the aromatic ring, depending on the aromatic ring, because orthonitropentaloxycarbonyl is common and then the linker is also common. So, what is different is the substitution pattern in the aromatic ring. So, you add T2 and then here you add suppose serine, but when you add serine you do not need to add another tag T3, but what you can do, you can add T1 and T2 both. So, what will happen here, the bead will have glycine and then T1, and this bead will have alanine and T2, and this bead will have because there are different valency sites for the tagging. So, tagging sites are also quite a few on the bead. So, you will have T1, either T1 attached or it could be T2 attached to some of the sites. So, in case of serine you have T1, T2, in case of alanine you have T2, in case of glycine you have T1. Okay. Now, suppose I stop here, I mix it, I mix the beads and then I distribute, that is the mix and split synthesis. So, I can now mix it and then split it into 3, okay. And then I add another glycine. So, in this port what will happen, because I have mixed all these, so it will have glycine, and whenever I add glycine, what I do, I add T1. So, whenever there is glycine it will only have T1. Whenever, because this is mix and split, again I repeat, each port now will have all the 3 components. So, I will have alanine here and this alanine will be attached to glycine, but the because I am because I have added your, so T2 is already attached to the alanine bead, and the other sites now I have, whenever I add glycine I add T1. So, that will have T2 and T1. Okay. And then I have the third one, that is serine. So, I added glycine. So, whenever I added glycine, because serine already has T1 as well as T2, now I have added glycine, the glycine I add only T1. Okay. T2, T1, let me see just whether it is correct, T2 and T1. Okay. Now, you see, if you stop at the stage 1, you have mixed it and I want to know each bead has what, whether it is attached to glycine, whether it is attached to serine or whether it is attached to alanine. How do you decide? You separate the bead and then strip off this, you shine light. So, what will happen? These T1, T2s will fall off containing different aromatic rings. Now, you push it into the, your HPLC chromatogram. If you put it into the HPLC, you get a chromatogram, and then you match with your, you have a reference one, that where the retention times of each of these tags are there. So, what will happen now? Suppose a bead is showing, I stopped only first stage, means glycine. Question is whether the bead is attached to glycine, or it is attached to alanine, or it is attached to serine. How do I know? I just strip off this T1, T2, all these tags, and then push it into the HPLC or GC. So, what will I will get the different that peaks for the corresponding to this T1, T2, T3. Okay. And what happens that in the, if the bead is attached to only glycine, I will see only T1, because whenever I added glycine, I had only T1. If I see that there is T2 coming out as exemplified or as demonstrated by the HPLC, then I know that the bead is attached to alanine. If I see that both T1 and T2 are coming, then what will happen? If both T1 and T2 are coming out, then I know that it is serine. Okay. So, basically this process allows you to combine this T1, T2 and T1 plus T2. You are not you are not adding another extra one here, T3. So, that means if you want to discriminate between three reactants, three substrates, in this case glycine, alanine and serine, you need two tags. One is separate tags for glycine, a separate tag for alanine, and a combined tag for the T1 plus T2 for the serine. So, in the next one, when you do the, if you carry on the synthesis for glycine, you are adding a T3 now, you introduce T3, another tag. So, whenever you are adding the second glycine, you add T3, when you are adding the second alanine, you are adding T4, when you are adding the serine, you are adding T3 plus T4. Okay. And if you make a tripeptide, then when you add glycine, you add T5, when you add alanine, you add T6, and when you add serine, T5 plus T6. Okay. So, that means you have made now how many compounds? You have made 9 compounds, but you have used only 6 tags. You are not using 9 tags, okay, because you have a combination of tags. So, now suppose I see that in the bead I strip off all these whatever is attached as the tag, and suppose the tags attached are here are T1, T2 and suppose T1, T2, and I also see T3 and I also see T4. Okay. So, I stop at the T3 and T4. Now, I strip off these, I take the HPLC chromatogram, and what I see, that I could see T1, I could see T2, I will see T3 and I will see T4. Okay. So, in the so if I see T1 and both T2, because they are involved only in the first step of the synthesis, that means it must be serine, the first amino acid, because if the first amino acid had been glycine, I would not have seen the T2. If I only see T2, then I know that the first amino acid is alanine, okay, but I am seeing T1 plus T2. If that be the case, that means the first amino acid is serine. What about the second amino acid? The second amino acid, I will see what are my tags, T3 or T4 or both T3 and T4. So, here I am seeing both T3 and T4. If that be the case, that means the second one is also serine. So, that means I am having serine linkage. Okay. And so, now I can develop a problem like this. Suppose I see T1, I do not see T2, I see T3, I am making the tripeptide now, and then I see T5 and T6, T5 and T6. So, what is the sequence of the amino acids? In the first one I am seeing only T1. Now, T1 is given only when glycine is there, okay. And then T3 second one, the possibilities are that I will see only T3 or I will see only T4 or I will see T3 plus T4. Here I see only T3. So, it must be glycine attached to T3 is given for another glycine, okay. And then I see T5 and T6 together. If I see both together, that is the third step, that means it is serine. So, that is the sequence of the compound attached to a particular bead. Okay. So, this techniques actually simplifies. If you increase the number of amino acids, but proportionately you are not increasing the number of tags, because what you have done, you have basically it is a, if you say binary system, it is a binary system that if you have 0 and 1, but you can get a combination of, you can have only 0, you can have 1 1 or you can have 0 1. So, basically very similar to that. If you have on the other hand, suppose if you have T1, T2, T3, T4, T5, T6, then what will be the sequence of your peptide? It will be T4, T5, T6. So, I get T1 plus T2, that means it is serine. I get T3 plus T4, that means this is also serine, and I get T5 plus T6, that means that is also serine. So, that is the peptide, tripeptide. Okay. I think combinatorial chemistry, but there are I tell you there are other methods of doing this. I just mentioned two methods. One is this primer based method, where you add the DNA based sequences attached to the as the tagging, and then finally, you add the primer and then see which primer is hybridizing with the nucleotide sequence that is attached to a bead as the tag. Okay. And the second one, you are taking a photolabile orthonitrobenzyloxycarbonyl attached to through a linker to an aromatic ring. These aromatic rings are differently substituted. Okay. So, differently substituted aromatic rings. So, they will have different retention times. So, that acts as T1, T2, T3, T4, T5, T6, and the trick is that you use 2 tags for 3 components, that reduces the number of tags, because after all that will be expensive. If you need the similar number of tags, so that will have more, that is economically more expensive than the than if you have the reduced number of tags. Okay. So, that takes care of the combinatorial chemistry and the associated techniques. And so, next we will go to the some medicinal aspect, but before that we have now discussed the drug discovery process in general, what is done, what are the different steps, how you do that, and the second thing that we did is the combinatorial chemistry, that is the requirement for the day, that how to get a large library of compounds and then get it tested very quickly, and also how to know the structure of the compound that you are generating and that is attached to a particular bead. Okay. Thank you.